All right, now let's move on to the cells of the immune system, including lymphocytes.
When we talk about lymphocytes, we need to talk about first the two main branches of the immune system. So there's the innate immediate immunity, and then there's the adaptive or delayed immunity that comes later. Let's talk about both of them real quick. So just a brief comparison, some general facts about the innate immune system. Like I said, it's the body's first line of defense, very rapid acting and non-specific response to pathogen or disease. It's non-specific, but the immune cells use these TLRs, or toll-like receptors, to recognize PAMPs, and PAMP stands for pathogen-associated molecular patterns. So these are just general bacterial and pathogen products, viral or any type of pathogen products that the toll-like receptors recognize on the immune cells, and they just see these, they recognize them as foreign, and then they attack. So that's how that works. Doesn't need to be specific at all. Whereas the adaptive immune system is delayed because it is specific. It takes time actually for the genes to recombine and rearrange themselves and be made exactly perfectly complementary to whatever pathogen is invading the human body. So this is called VDJ recombination. And we'll talk about it a little bit later in more detail, but this is how antibodies develop and become exactly complex to an appropriate pathogen. They're perfectly made, like a puzzle piece connecting to another puzzle piece. All right, so the cells of innate. Neutrophils, do you know any other types of cells? So again, neutrophils, they just release, what do they release? Reactive oxygen species. Okay, so all kinds of free radicals and volatile oxygen derivatives that combine with pathogens and just tear up and lyse their membranes. Very nonspecific. Even they'll lyse any type of membrane. If they come in contact with even your own cells, they'll destroy those too. So the neutrophils try to direct them towards the pathogens, but not always. We need to have special enzymes that break these down in our body so that our own cells don't get hurt too badly. Okay, so the neutrophils, what else? Eosinophils. That's right. What type of hypersensitivity, we'll talk about this later, do we see eosinophils? Type 1 hypersensitivity, so allergic responses. That's an immediate or innate immunity response. All right. And then adaptive cells are like macrophages. Those come around. Those are more specific and specialized killers. Dendritic cells, we talked about those a little bit earlier. Remember, they come from tissues that come into contact with the outside environment. And then they bring a piece of pathogens to lymph nodes and tell the lymphatic cells to get going and get making things to combat the pathogens. All right, so T cells, those are specific, and B cells too. They can be actually innate and adaptive. If they're making IgM, that's the first type of antibody that they make. See, it's a pentamer. Then that is associated with the innate immune response. And I think of IgM as immediate. That's a nice mnemonic there. But if these B cells class switch their antibody, we'll talk about how they do that later, and start making IgG, that is more associated with the adaptive arm of immunity. Okay, so got the overall major differences between these two types of immunity? I hope so. We'll talk about them a lot more in detail. I just wanted to provide you with a general overview right now. All right, now it's time to talk about how cells grab onto antigens and process them and then present them to other cells so that they can stimulate the other cells. So, they do this through MHC receptors, okay? So MHC stands for Major Histocompatibility Complex, and there are two main types. We'll talk about the two main types, starting with the first, MHC1, okay? And this is really associated with every type of cell in the body, actually, except for one. Do you know what cell does not possess an MHC1 complex? Red blood cells. Very important to know. Erythrocytes do not have MHC1, all right? So, what happens is, if, let's say, a sample cell, a poor cell, is infected with a virus, every cell, except for red blood cells, have proteasomes that then degrade the virus, process fragments of this virus, stick them in the rough endoplasmic reticulum, and then a fragment of this MHC1 receptor, do you know what fragment that is, brings it to the surface. What fragment brings it to the surface? This is important, actually, for boards. The beta 2 fraction, okay? You gotta know that. And actually, because it's important in patients who are on dialysis. Why? What disease could patients on dialysis get as a result of beta 2? These beta 2 fragments do not get filtered by dialysis, and they can actually accumulate and cause amyloidosis, or an accumulation of misfolded proteins in your organs. Ooh, that's a board question. Alright, so anyway, this fragment of virus is stuck onto the surface of an MHC1 receptor. And this is like a warning sign saying, hey, I'm infected with a virus, help, 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 I'm infected with a virus. And this notifies other types of cells that we'll talk about later that can go and complex with this receptor and kill this cell. Or tell the cell to kill itself, actually. So that's how that works. We'll talk about it more later, but let's first discuss the MHC2 receptor. So what takes place here? MHC2. All right, so here we have a macrophage, or in general, MHC2 receptors are on all antigen-presenting cells, like macrophages and actually B cells too. And you think of another type of cell? Dendritic cells as well. All right, so what happens is inside the macrophage, there's a phagosome, okay? Phago means consuming or eating. So an organelle that will consume, let's say this macrophage just ate, was hungry, and just had a nice snack, nice bacterium snack. So it goes in the phagosome, and then this lysosome, which has all these catalytic and degrading enzymes in it, combines with the phagosome, and it's called, well, this is creative, phagolysosome. So this organelle now has a bunch of reactive enzymes in it that will tear up this bacterium and process it and degrade it into small pieces. So then comes along the MHC2 complex, which is brought to the surface and to this phagolysosome by the invariant chain. For some reason, boards might think that's important, but it helps transport the MHC2 receptor. It's like a chaperone. And once a piece of this bacterium gets stuck onto the surface, the MHC2 receptor is brought to the surface of the macrophage, where it can be presented to other types of cells. So that's the MHC2. All right, so you got it? Got the MHC receptors? They're very important. So now it's time, this is an easy question. What receptors mediate immune and non-immune cell interactions? So, just talked about it. This is just to make sure you got it. The MHC receptors. Very good. So, okay, real quick. What MHC receptor was present on antigen-presenting cells? 2. What about on every type of cell except for red blood cells? 1. Very good. So, continuing on with MHC receptors, it's very important and very high yield to understand the differences between them. So, the HLA genes are actually the genes that are responsible for coding for the MHC receptors. And MHC1, do you know what HLA genes code for this? HLA-A, B, and C code for that one, okay? Then for 2, how about that? What genes code for that? It's kind of fancy here. DP, DQ, and DR. So ABC and pretty much PQR, okay? That's what you need to remember. And you can think of the MHC2 as having 2 letters that code for each one. So like the DP, that's 2 letters. Kind of helps you remember it. So let's go over some of the main types of these HLA genes that are associated with diseases. Some people have certain HLA genes that make them more susceptible to certain autoimmune diseases where their body attacks themselves. Okay? So how about for the A3 gene? It's associated with hemochromatosis, okay? Just gotta know that. A3 is hemochromatosis. How about the B, and specifically B27? What disease is associated with that? Well, I remember the mnemonic PAIR. It's in their first aid book, but it stands for, do you know? Psoriasis, ankylosing spondylitis, inflammatory bowel disease, and Reiter syndrome. So I'm not going to get into the details of all these diseases. You're going to talk about them later, but just know what diseases are associated with what genes. It's just a memorization thing. All right, let's move on to some MHC2 receptor and HLA genes. So the DQ2 and DQ8 is a good one to know. What disease? Celiac disease. All right, very good. And then finally DR4, last example. Remember that this is rheumatoid arthritis. So just, I know it's pretty much rote memorization, but come back to this once you've studied it and test yourself. So let's now get into some of the specific types of cells of the immune system. And we'll talk about a fun-sounding one called natural killer cells. Ooh, they're natural-born killers. Wow, sounds scary. So, when we talked about MHC receptors, we said that for the MHC1 receptor, there were certain cells that would complex with this and tell that cell that if it's virally infected to kill itself, right? So again, we had this virally infected cell where the virus was degraded and antigens were processed and placed onto the surface of an MHC1 receptor by what fragment is this? Beta 2 fragment, okay? And now we're talking about the cells that will complex with this. So one of those cells is a natural killer cell that can do it. So here's an NK cell, all right? It realized that this cell is infected with a virus. And it perforates a hole then through the membrane using an appropriately named protein called perforin that perforates, and then deposits what? What do you think it deposits inside? Do you know? Granzymes. Granzymes. They're enzymes that will stimulate what inside this cell? The release of something from the mitochondrion, and that is cytochrome C. So cytochrome C gets released. And that's not good because cytochrome C then in turn is going to activate something that starts with a C, and that is caspase enzymes, and those lead to apoptosis. We'll talk about this in the inflammation chapter, but apoptosis, I think of a cell popping because it is basically cell suicide. The cell shuts down and tells itself to die, basically. Programmed cell death in a nice, neat, organized fashion without any inflammation, whereas there's another type of cell death that has a lot of inflammation. What's that? That's necrosis, but we'll talk about that in the next chapter. So, apoptosis takes place, all right? And the cell fades away because the natural killer cells told it to kill itself. So, not only do they actually tell virally infected cells to die, they can also tell what other types of cells? What do you think? What can go wrong with a cell? Malignant cells, cancerous cells will be destroyed as well by natural killer cells. Okay, so moving on to other types of immune cells, we're now going to talk about the key players of the immune system, and those are B and T cells. Very, very important. Okay? Very important. So, B and T cells. B cells first. What do they do again? Remember, they turn into a certain type of cell. What is that? Plasma cells. And then they make, what do they synthesize? Antibodies. So, IgM first. This is not IgM. This would be later on. This is IgG, or could be another type of immunoglobulin. But what do these antibodies go and do? Well, IgG and IgM can go and activate the complement cascade. We'll talk about that later. Okay, that's another part of inflammation, another inflammatory pathway. Or, if this is IgE, it can go and stick on the surface of mast cells and participate in the activation of a certain type of hypersensitivity. What type is that? Type 1, the allergic response. What other cell type will we see there, probably? What other cell will be called in? Eosinophils, right? Right. Okay, so T cells, what do they do? Well, there's two main types, two general types. Helper T cells and cytotoxic T cells. So helper T cells, they do what their name is. They just help everybody, okay? They release cytokines that go and do a number of things, stimulate all kinds of immune cells, even B cells. They cause B cells to class switch. They cause macrophages to kill bacterium that are inside of them. So they also stimulate themselves. They stimulate other types of T cells, all right? And they can even inhibit other types of T cells. So, but in general, they help. They help the immune system through cytokines. That's what helper T cells do. Cytotoxic T cells, they bind to the MHC1 receptors just like natural killer cells do, and they kill virally infected cells and neoplastic cells through what process was that called? Through apoptosis, right? Remember? Ring a bell? We just talked about it. So that's a broad overview of B and T cells. Now we'll get into some more details, all right?